Okay.
I'll try not to cough in your video.
Right? All right, hopefully you can see enough and it'll be loud enough to where it makes sense. All right, so again, oh, I could tell you're already watching. That's good. You found it. So here's the thing. I know there's a lot of slides. Whoa, what is that? Oh no! Oh, by the way, if you haven't grabbed the cupcake yet, please feel free to grab one. So let's go ahead and start. I know there's a lot of slides, so I'll try to focus on the things that you need to look at, you know. I'll also try to point out anything that you don't need to focus on. But at the same time, try to learn the material on these slides, right? Because I might miss something, and I don't want to miss something. Yes, feel free. Let me see here. With microbial growth, what we should realize is we need a source of carbon, oxygen, nitrogen, hydrogen. There are macronutrients, mainly C, H, and O, right? And then there are trace elements as well that we need to live. They're called micronutrients. Now, do you remember we need to figure out sources of carbon, hydrogen, oxygen, and electrons for these creatures, and energy for these creatures? In fact, that's how they get these like convoluted names, these three-part names. You remember these three-part names? So what I would do is learn this table here. I would learn this table. So the difference between an autotroph and a heterotroph, that tells you where a creature gets its carbon from. The difference between a phototroph and a chemotroph, that's where it gets its energy. And the difference between a lithotroph and an organotroph, that's where it gets its electrons. Yeah.
Could you raise the screen up just a little?
Oh, yeah, because it's dark at the top. That's a little better, right?
Yeah.
Yeah. So know these terms, because I might have like a question where it's litho, organo, heterotrophs get their electrons from where, you know, or something like that. So those types of questions. So do know that table there. Know the difference between an aerobe and anaerobe. What is an obligate anaerobe or an obligate aerobe? We talked about oxygen requirements. Remember, it is a feature to live in the presence of oxygen. It is not an adaptation to be able to live in the absence of oxygen. Life lived for quite a period of time before oxygen was available. Remember? And these are the enzymes. These are the enzymes that allow creatures to live in the presence of these free radicals, in the presence of this highly reactive oxygen species. Okay. Again, know what microaerophiles are, facultative anaerobes, aerotolerant anaerobes. Okay, and again, the ones that can live where there is oxygen, those ones have these enzymes, right? That's why they can live in the presence of oxygen. We talked about nitrogen, we talked about nitrogen. You remember, you could get nitrogen from nitrite, nitrate, you could get nitrogen from the air, that's called nitrogen fixation. Creatures need a source of phosphorus and sulfur. And of course, the different vitamins and such. What else did we talk about that was important? We talked about, oh yeah, these are the cardinal temperatures, right? Growth temperatures, the minimum, optimum, and maximum temperature for a creature. What is that? Again, every creature has its own minimum temperature that it can grow at, optimal temperature that it prefers to grow at, and maximum temperature that it can grow at before it starts to denature, right? Before it starts to die. Psychrophiles like it cold, right? You don't need to know the exact temperature range, right? Just know psychrophiles are creatures that grow in the coldest conditions. Mesophiles grow around body temperature. Thermophiles grow, warmer than body temperature. And hyperthermophiles grow basically in boiling temperatures, very, very hot temperatures, like thermal vents and stuff. But again, you don't need to know all the exact temperatures. We talked about pH, remember? Alkalinophiles, acidophiles versus neutrophiles. So do know those terms. Physical requirements. What did we talk about with physical requirements? Oh, remember hypotonic versus hypertonic? You know, you might want to brush up on that. Hypotonic environment means high water concentration outside of the cells, right? Like pure water. You know, like if you put a cell in pure water, that's hypotonic condition. If you put a cell in salty water, for example, that would be a hypertonic condition, and water would leave the cell. Barophiles, these guys withstand pressure. What else did we talk about? Quorum sensing. Remember what quorum sensing is? It's a strategy of communicating with other bacterium by releasing this quorum molecule, right? It's a way of figuring out how many bacteria are in the environment. And what are examples of quorum sensing? Making plaques, right? Making plaques is an example of quorum sensing. But you don't need to know every step in the plaque formation process. You don't need to know all of this. Or this, or this. Now with binary fission, remember this is how prokaryotes, well not all prokaryotes, but most prokaryotes divide by binary fission, right? Remember the steps to that? You've got a circular chromosome. Usually it's one circular chromosome. The DNA starts to get copied at one particular point called the origin of replication. And it gets copied in both directions until you hit the termination site. Now you have two copies of that chromosome, that circular chromosome. They separate to opposite poles of the cell, and then what happens? The cell wall starts growing inwards. That's called septum formation, and you form two cells, right? So it's very straightforward. Yeah.
So know that.
Yes. Yeah. All right, next thing. This here generation doubling time, you don't need to know these equations. It's just showing you how, you know, cells divide exponentially. And a bacteria cell, for example, E. coli will divide every 20 or so minutes, right? So that's why you can end up with millions or billions of cells within a day or two. But you don't need to know the exact equation. But you should know it's a logarithmic, it's an exponential, logarithmic scale growth rate. Now you do want to know in batch culture, remember the phases of growth? You should know them in order and kind of what's happening during each phase. There's a lag phase, logarithmic or exponential growth phase, stationary or senescence, stationary phase, and then senescence or death. Senescence or death. In that order, right? In that order. Okay. Any questions about chapter 6? No?
So you said being able to live in oxygen is a feature?
Yes, yeah. Being able to live where there's oxygen, creatures need specialized enzymes that help them cope with reactive oxygen species, right? If you don't have these genes to make these enzymes, then oxygen actually tears you apart. Oxygen destroys the cell. Yeah, did you have one?
The heterotroph and the autotroph, that's table 6.2?
Yes.
6.2.
Yes.
What exactly should we know from that table?
Basically, what's on the, hold on, let me show you. So for example, I might give you the term chemolithoheterotroph and ask you, where does this creature get its electrons from? Well, what would be the answer? Yeah, reduced inorganic molecules.
Okay.
Does that make sense? So remember, this is a three-part word, right? These are three-part epithets, right? You've got creatures, and you can categorize them based on their dietary requirements, right? Where are they getting their carbon? Where are they getting their energy? Where are they getting their electrons? And that's what makes up the three-part word, right?
So we're looking for the energy source?
It could be energy source, it could be electron source, it could be carbon source,
Okay.
right? Because the terms autotroph and heterotroph have to do with carbon source. The terms phototroph and chemotroph have to do with energy. And the terms lithotroph and organotroph have to do with electron source. Yeah. So for example, a photolithoautotroph, right? That term, this three-part term, tells you electrons, energy, and carbon source. Yeah. Yeah. So basically, know this table. If you know this table, you could answer any of these questions about these organisms. Yeah. Any other questions about chapter 6?
No, the question she asked you about,
Yeah.
Oxygen.
Yeah.
When you explained it, they had a follow-up sentence.
Yeah, you
said
talked
you
about
said-
adaptation.
Yeah, if you live in oxygen, it's a feature. It's a
Then
feature.
if you don't live, if you're not able to live in oxygen, is what?
That means you don't have certain enzymes that you need to neutralize reactive oxygen species, right? Even you and I need, catalase and stuff. We need enzymes that help break down those reactive oxygen species. If we didn't have those enzymes, we would get torn apart by oxygen and we'd die. Yeah. Oxygen is very toxic stuff. It's very oxidative, right? That's where the term oxidative came from. Oxygen's ability to steal electrons from things. And so oxygen is very toxic. And so creatures that live where there's oxygen need special enzymes that help them cope with oxygen. Otherwise, oxygen starts stripping electrons from everything until the cell's dead or the organism's dead. Yeah. It's pretty weird. Think about it. Isn't that like Twilight Zone weird stuff? We're living in the waste products of plants, and we need it to live. And it's kind of a toxic waste product, actually. Oxygen is very strange. Yeah. That's spooky stuff to know for Halloween. Very scary. All right, so anything else? No, not on this chapter, right? Next chapter? 9 and 10. Let's go. Whoa. Let's see what's going on with 9 and 10. All right, 9 and 10. Microbial growth. Do know all of these terms, right? Sterilized versus disinfected. Sterilized means you've destroyed any viable cells, right? Any viable pathogens or things like that, right? What about disinfect? Is it astringent? No. No, disinfect means you've killed most, okay? So know that. Sterilized means you have destroyed all possible viable cells, right? And disinfect means, 99.99, it's like Lysol, okay? It's not as stringent. Sanitation is the least stringent, right? It's just getting levels, reducing levels to acceptable for health level, levels. Antisepsis is treatment on tissues. So mouthwash is an antiseptic, right? Chemotherapy is ingesting or bringing in some chemicals that help reduce microbial growth from inside, right? Like an example is what? Antibiotics. Antibiotics. Remember that cidal agents kill. Cide means kill. Static means just prevent the growth. Yep. Do you remember decimal reduction time? That means the time it takes to kill 90% of these creatures. And different disinfectants are going to have different decimal reduction time depending on these things, right? Population size, population composition, concentration of the agent, duration of exposure, temperature, and local environment. These types of things can affect decimal reductions. They can affect how strong the, or I should say how potent the agent is.
So we should know.
Yeah, at least be aware of them. And then we have moist heat. Moist heat, we have boiling. Does that sanitize, or, I mean, does that sterilize? No. But this does, right?
Yeah.
Autoclaving sterilizes. Why? Because you're increasing the pressure, and because you're increasing the pressure, it's like a pressure cooker, you can increase the temperature. You know when you boil water, it never really gets past 100 degrees C, right? Does that make sense? You know why? Huh? Evaporates, right? It starts evaporating, right? So all the really hot stuff just bubbles its way to the top and evaporates, right? Does that make sense? So you can't get, at home when you're boiling, water, it might be boiling really fast, and you might think it's, wow, this is really, really hot, but it's, no hotter than, any other boiling water. Boiling water is 100 degrees C, right? And so that's why it just doesn't get that hot to kill spores and things, right? But if you put a pressure cooker, if you put a lid on it, right, like a pressure cooker, now it's getting hot and it wants to, evaporate, right? Can it evaporate? No, it's stuck in there. In the pressure cooker, it can't evaporate, so it stays liquid, but it's just getting hotter. You're not allowing it to evaporate. You see what I'm saying? So it can get 120 degrees Celsius, for
That's what an
example.
autoclave does?
What an autoclave does, yeah. So it still steams, but the steam's kind of pressurized. Everything's kind of in there pressurized, and the pressure allows you to go past 100 degrees Celsius, right? Does that make sense? And that's strong enough to kill spores. Pasteurization. What is this?
Heating below boiling.
Yeah, heating below boiling, just to get rid of most pathogens, most bacteria. Dry heat, this would be an incinerator or fire or something like that. This can sterilize if you get hot enough.
It can?
It can, yeah. Like fire or an incinerator. We're talking about, really high temperatures here over a long period of time. Okay. Now, what's the drawback to filtering? Viruses, yeah, viruses are really tiny. So a lot of times viruses can make their way through the filter. Filters can be pretty good against bacteria and stuff, but not always great against all viruses. And then more filtering. UV radiation, obviously, that kills bacteria, but what's the drawback there?
It cannot get into dirt.
It can't get into dirt or water, or remember even that little piece of plastic, the little petri dish cover was enough to stop UV. So, UV is pretty strong. If the UV hits the bacteria dead on, yeah, the bacteria dies. You guys saw that. But if it's like hidden underwater or under a dirt, or if it's sheltered by even an index card, right, it's fine. So that's not good. Radiation's really good, isn't it? Radiation, especially if you're dealing with stuff that's heat label, right? Things that can get denatured by heat. You could use radiation to kill the bacteria and things. It's not 100% against viruses, but it's not a big deal. What else? Okay, so now these are agents, chemical agents, right? What do I want to know? You don't need to know all of these chemical agents. You don't need to know the difference between these agents, right? But do know that phenolics include phenol and triclosan. Those are two big ones to know. Phenolics, phenol, and triclosan. Why? Because you'll probably encounter those quite a bit. Okay? And triclosan is found in hand sanitizer. You may want to know that. And what else do you want to know about phenolics? That they have a ring structure to them, right? That's a good thing to know. Phenolics have a ring structure to them. Okay. I'm not sure if I ask anything about this, but it's very good to understand that overuse of antimicrobials is leading to more resistance. Why? Because you're killing off the less resistant microbes and leaving behind the more resistant microbes to fill in that, niche, fill in that real estate.
But you said it's not in the.
I don't know for sure if it is or isn't, I forget. So it might be worth knowing. Alcohols, just know, ethanol, isopropanol, right? These are pretty strong disinfectants. The halogens, which ones do you need to know about? I would want to know about chlorine and iodine. Okay. Heavy metals, right? These are toxic to microorganisms. Quaternary ammonium compounds, I would want to know that these are kind of like disinfectants and wetting agents, etc.
The what?
Disinfectants, I'm sorry, not disinfectants, detergents is what I meant to say. Detergents, wetting agents, things like that. Emulsifiers. Aldehydes, you might want to know about formaldehyde and glutaraldehyde. They cross-link proteins, they cross-link things, and so they're very strong disinfectants, if not sterilants. They can even be sterilant. They cross-link, yeah. Yeah, they're good for cross-linking proteins and cell components. Okay. Sterilizing gases, you don't need to know the different sterilizing gases. Just know that sterilizing gases are gases that can be used to sterilize, right? You don't need to know about all the different examples there. They're kind of easy to forget. Okay. So any questions about chapter 9? No. Now let's move on to chapter 10. Remember, this is chemotherapy. Chemotherapy, to a microbiologist, we're talking about regulating microbial growth inside, right, by administering drugs. So we're talking mainly about antibiotics here, right? You should know these terms here in bold. Selective toxicity, therapeutic dose, toxic dose, therapeutic index. Is a high therapeutic index better or a low therapeutic index? Higher therapeutic index. What that means is you can ingest more of the drug without having side effects, without having the problems, right, associated with it. What that means is there's a big difference between the therapeutic dose and the toxic dose, right? Just know these terms. Anything in bold, just learn the terms, right?
Just the whole slide.
Just the whole slide. Yeah. And then now, okay, here we go. We're talking about different types of antimicrobial drugs, different types of antibiotics. You should know that some antibiotics act on the cell wall, some act on protein synthesis, some act on metabolism, and some act on nucleic acids, right? So penicillins, I would want to know about penicillins. I want to know that they act on the cell wall, right? On the cell wall. And what else do I want to know about penicillins? You don't need to know all the different penicillin types. It's good to know, but you don't need to know it for the exam. What do you need to know? Well, first of all, penicillin has this beta-lactam ring. Remember this little ring structure? And that's a weak point, right, for penicillin, because some bacteria can make an enzyme called beta-lactamase. And that enzyme, like scissors, comes in and chops up that beta-lactam ring, and that renders the penicillin useless, right? So bacteria that are penicillin resistant are able to produce an enzyme which chops up the little lactam ring on penicillin. And so that's why you have things like methicillin, which is kind of like penicillin, but it's resistant to that beta-lactamase. So it's good to understand those types of concepts are probably important for you guys to understand. You don't need to go into tons more detail than that, right? Yeah, again, I don't really go into any of that stuff here. This is just interesting information for you, but you don't need to dwell too much on it. Cephalosporins, I don't even think I ask anything about this, but just know, if there's anything to know, is that cephalosporins are another type of cell wall inhibitor, right? Cephalosporins. And vancomycin is another type of cell wall inhibitor. That's, what to know there. You don't need to be able to identify it or pick it out or anything like that. And you remember this used to be, vancomycin used to be the antibiotic of last resort, right? It was the antibiotic they, broke the special glass container to get out, if nothing else treated the infection. But nowadays it's really overused as well. And then we talked about protein synthesis inhibitors. Okay, protein synthesis inhibitors. So what you should know about tetracyclines is, this one might be good to know. It's a, tetra that has 4-ring structure, and it's effective against protein synthesis. You don't need to know exactly that it combines with the 30S ribosomal subunit. I mean, that's getting too detailed. It's getting really, really into the woods there, right? Doctors don't even know, when they're prescribing medication, they don't know 99% of the time what is the exact drug target of this particular drug. So, you don't need to go into this. This isn't a pharmacology class. Now, if this was pharmacology, I'd care more that you know the exact drug targets. But obviously, these drugs target what? If they are protein synthesis inhibitors, they target what? Yeah, ribosomes, or maybe even mRNA after it's made, or tRNA, or something to do with the ribosome. I don't know. Does that make sense, right? And if it's a cell wall inhibitor, what does it target? Those drugs target? Yeah, components of the cell wall, or proteins that maintain the cell wall or help the cell wall build itself. I don't know, right? Does that make sense? Now you get how drugs work. Drugs are just small molecules that bind to and prevent the function of different parts of the cell, right? So anyway, again, you don't need to go through and say, oh, I need to memorize, that, this particular drug targets the 30S subunit, and this one targets the 50S subunit, and things like that. You don't need to worry about all that detail. So tetracycline is an example of a protein synthesis inhibitor. Macrolides are an example as well. Chloramphenicol is an example as well. You don't need to go into more detail than that, right? Just don't worry about it. Sometimes I put extra information on there because it is interesting and I want you guys to know about it, but not to sit there and memorize it, right? Metabolic antagonists. What are these? Oh, I got it for you. Metabolic antagonists. These usually are drugs that mimic important metabolic substances that organisms need, right? What was the example I gave you? Sulfonamides or sulfa drugs. In this case, the sulfonamide is mimicking this other guy, P-aminobutyric acid, which is necessary. Remember this PABA, right? PABA is necessary to make folic acid. You need folic acid, right? Well, the sulfa drug mimics PABA, right? So what does the bacteria do? It'll use that sulfonamide to try to make folic acid, and now it's making this dummy molecule that it doesn't need, it can't use. So it just wasted a bunch of resources, time, effort, right, energy to make something it doesn't need and it can't use, right? Does that make sense? So that's how they work. You don't need to know the exact mechanism of how this works, but that's how metabolic antagonists work. Nucleic acid synthesis inhibition. What do you think these drugs target? Anything to do with DNA or RNA, like making DNA, making RNA, right? Yeah, I mean, you know what polymerase is? DNA polymerase is the enzyme that actually makes the DNA copies of DNA. You know what RNA polymerase is? the enzyme that actually makes mRNA, right, that actually makes RNA. So these drugs target those enzymes that are necessary to make DNA or RNA, right? So that's why they prevent. Okay, so just know that quinolones are an example, okay? And moving on, antifungal drugs. How come it's harder, this is a good thing to know, how come it's hard to find antifungal drugs that have a high therapeutic index, that are very potent against the fungus, but not you?
Because it's actually similar.
Eukaryotic.
They're eukaryotic, they're similar to you, right? Fungi are eukaryotes. Eukaryotes are more genetically related to you. It's harder to make drugs against their proteins because their proteins look like your proteins and their genes are like your genes. So, you know, unlike a bacteria where you could target their, you know, ribosomes, because their ribosomes look a whole lot different than your ribosomes, or you could target their cell wall because you don't even have a cell wall, right? Fungi, they really don't have all that many different structures that you don't have, right? Does that make sense? They're more closely related to you, and that's why it's harder to treat fungal infections, and that's why fungal treatments tend to be toxic to you as well, right? Does that make sense? Antiviral drugs, do these usually outright cure you? No, the antiviral drugs, they're designed to slow down the virus. Your body will usually cure yourself, or it won't, okay? Does that make sense? Like in the case of the flu, it'll slow it down, the Tamiflu will slow down the flu until your body has a chance to cure yourself, right? Does that make sense? In the case of anti-HIV retroviral drugs like cocktail, it'll just slow it down. You know, it just never gets cured. You know, it just depends. Can your body cure you or can your body not? Right? That type of thing. So again, with the HIV drugs, that's what a cocktail is. A cocktail of drugs means you're ingesting a medication, but it's multiple drugs in one. It's multiple drugs in one. You're targeting different proteins in that virus's cell cycle, or life cycle, I should say. Right? Does that make sense? You don't need to know all the different targets, but just know that antiviral drugs aren't just one drug. It's multiple drugs with multiple targets, and that's why it's really good at slowing down the viral replication. Okay, and then these are factors influencing antimicrobial drugs. This is just a list to understand. What else? MRSA and VRSA, remember vancomycin resistant and methicillin resistant Staph aureus. It's good to just know about them. Oh, these are mechanisms of drug resistance. You may want to know these. These are strategies that microbes employ to be resistant to drugs, right? And that's that. Any questions about these two chapters? Okay. Moving on. Let me see. Okay. You know what? Maybe I have some good news for you.
Screw this whole chapter.
No, not that. Not that. We'll see. I'll let you know. Okay, so moving on, we were learning about prokaryotes. We did a survey of prokaryotes. We started with the archaea. Remember, archaea live in extreme environments, extreme pH, extreme temperature, right? Things like that. That explains why they don't cause a lot of human conditions, right? Because you're a very mild, you know, you as a mesophile and all these things that archaea just don't want to grow on your body, right? So that's why archaea really don't cause human disease. Now there are two phyla, right? You should know there's two phyla of archaea, okay? Euryarchaeota and Crenarchaeota, okay?
Do we need to know this breakdown?
Well, this is what, right now we're just talking about these two. The Euryarchaeota and the Crenarchaeota. Those are the two phyla of archaea, okay? So, do you remember the difference? The Euryarchaeota can be any of these types of archaea. Remember this? Some of them are methanogenic, some of them are sulfate reducers, some of them are halophilic, some of them are cellulose, and some of them are sulfur metabolizers. Right? But what about the Crenarchaeota? Yeah, they can only do this. They can only, reduce sulfur and their thermophiles. They're only that one category. So that's what you should realize. You should realize that the Crenarchaeota are these thermophiles that reduce sulfur, and that's it. They're one-trick ponies, right? That's the one thing they can do, okay? Whereas the Euryarchaeota are way more diverse. They can do all of the above, right? That's what we need to know about them. Then we moved on to the gram-negatives, or at least some of the gram-negatives, the dinococci and the non-proteobacteria gram-negatives. You see everything in color here? We talked about them, everything in color. So what do we need to know about Aquaficiae? Oldest, right? Thermatogae, second oldest. Dinococcus thermus, resistant to desiccation and radiation. There actually will be questions just like that. Which one's the oldest? Which one's the second oldest? Which one, is most resistant to desiccation and radiation? So that's like 3 free questions right there. Chlorobi, just know that they are the green sulfur bacteria. That's all we need to know about them. That's all we need to know about them. Chloroflexi, green non-sulfur bacteria. That's all we need to know about them. Okay? Cyanobacteria, these are a little more interesting. These are the biggest group of photosynthetic creatures on the planet, right? They fix CO2. They take CO2 out of the air. They're great at reducing greenhouse gas. Take CO2 out of thin air, right? And produce oxygen for us to breathe. They're really important, right? Chlamydiae. Chlamydiae. What about these? Includes the genus Chlamydia. What's interesting about that? It causes chlamydia. It's the causative agent for chlamydia, right? They're obligate intracellular parasites. Obligate intracellular parasites. That's what we want to know about them. It's spread by sexual contact. Okay. What else did I want to know? Oh, I want to know the life cycle of chlamydia. Okay. Remember? 1. Elementary body, which is the external protective form of the bacteria, attaches to a cell, one of your cells. It triggers endocytosis. Then once inside, it change to an RB, converts to a reticulate body, right? From that, it can divide, reconvert to elementary bodies, and eject out of the cell, trigger exocytosis, and infect the next cell, right? So do know the life cycle of chlamydia. Okay. As far as the infection, like how it actually infects, I'm not as concerned. Now, what you should know is chlamydia can cause genital infection as well as eye. And the eye infection, you should know, is called trachoma. Okay, so you should know that chlamydia can cause trachoma in the eye or genital infection as well. That's about it. You don't have to go into tons of detail there. Right? Spirochetes, right? Phylum Spirochates. What about this? They have a helical shape. Why do they have a helical shape? They bore, they have the axial filament that helps them to corkscrew or bore into mucus and things like that. They can cause Lyme disease. Some of them cause, one of them causes syphilis, right? Okay. The one that causes syphilis, Treponema pallidum. What do we need to know about syphilis? We should know primary, secondary, latent, and tertiary syphilis, right? What happens during each. During tertiary syphilis, is it really treatable anymore? No, because it's triggered a autoimmune overreaction, hypersensitivity, and now your body is causing damage to itself, and now it is untreatable. If you allow a patient to progress to tertiary syphilis, there's really nothing much you can do for them at that point, right? That's when they start forming these gumas, right? So you should know you start with a painless lesion, it then goes systemic to a body-wide rash, and then it can lead to gumas, these problems with the autoimmune, with their skin. Okay, so this was the surprise I had for you. Halloween, not scary surprise. This, I think I'll stop here for the next exam. Like this will be the cutoff point for this exam. And the rest of this, it's not that we're not going to do the rest of this. I'm just going to, I'm going to save that. I'll save the rest of this for the next exam. So we break it up a little more, right? Does that make sense, right? So less to really digest for the next exam. Does that help? Yeah. Okay. So this slide here, actually the next slide, that's the cutoff point for this next exam. Yes.
What's on the next slide?
No.
The
Yes.
pictures.
When they say neurosyphilis, is that considered tertiary syphilis, or is that just an infection of the brain with syphilis?
Neurosyphilis, that makes me think it's going like into the actual neurons, you know, it's actually infecting the neurons. That may not be what they're talking about, because the autoimmune.
So tertiary just means the autoimmune
Yes,
phase of it?
yes. Yeah, as far as I remember, yeah. All right, guys, so I'll stop the recording
You
here.
said the next slide is on?
Yes.
So chapter